There we go. Of course I forgot that. All right. If I ever forget that and you don't see the little red dot by my name, remind me that I need to be recording. Okay, so we're recording. We just talked about the functions of the muscular system and therefore of muscular tissue, which are all listed here. And we were finishing up by saying that heat production occurs because, muscle tissue is very metabolically active. Metabolically, there we go. And so whenever we are using a lot of ATP, we're generating heat. So the more muscle activity you have, the more heat generation is going to come from your muscle. And that is definitely why when you exercise, even on a cold day or in a cold room, you're perfectly capable of breaking into a sweat because you're still generating enough heat to need to cool down again. All right. Now, when we look at all muscle tissues, of which there are 3 types, they all share certain characteristics that distinguish muscular tissue from the other major tissue types. So the things that distinguish muscle tissue from epithelial tissues, connective tissues, and then nervous tissues are these characteristics. So muscle tissue is excitable, which means that it's capable of responding to stimulus. So it's capable of having a response or an action. The particular things that are quite unique to muscle are that muscle is contractile, which means it has the ability to actively shorten its length. And the things that go with contractility are elasticity, so the ability to bounce back into normal shape or length after being stretched, which is a character called extensibility. So this combination of characteristics makes muscle very good at contracting and then relaxing and stretching out again. Other characteristics of muscle tissue are that it is vascular, so it always has a good blood supply. And like epithelial tissues, it is cellular. So there's very little extracellular matrix. Pretty much the entirety of the tissue is cells connected directly to each other. And then the thing that gives muscle tissue its unique contractile feature is something called myofilaments. So we'll talk a lot more about myofilaments after we get through our different types of muscle tissue, but they are the specialized protein filaments within muscle cells that are responsible for allowing muscle to contract and therefore create movement. So a little bit more about these special features. So excitability is actually a quality that muscle tissue shares with neurons. So this is a quality we see in nervous tissue as well, which we'll talk about more when we get to the nervous system. The way that cells are excitable and able to respond to stimuli is by changing the charge of their plasma membrane. So we talked a little bit about this when we were talking about cells and transport of things by diffusion and by active transport. This idea that a membrane can have an electrical charge across it. And so by changing the electrical charge, they're actually transmitting electricity, and that electricity is able to stimulate the cell to have a response. So we'll talk more about how that works when we get to the physiology of the cells, but that's the basic concept behind excitability. So here's a little diagram showing you the types of cells that have excitability. So here we have a neuron sending an electrical wave, which we call a nerve impulse, that we can actually measure in volts. And here we have muscle that can also send an electrical wave that we call a muscle impulse. And so either way, we have this changing electrical current that goes down the length of the cell. And we'll talk Again, we'll talk more about that later. And then with contractility, I just like to reinforce the idea that contractility is this forceful deliberate shortening, and that you do need the extensibility and elasticity along with contractility to let muscle actually be effective. Because if all you do is shorten, but you get damaged when you get stretched out, or you don't bounce back really well, then you're not going to be as effective. So there's a cool little GIF of contractility as demonstrated by a heart. Give me 1 second. Get over here. Well, I tried. Kick some cats out. You know, you try to let them stay and they make a lot of noise. So, oh well. All right, so hopefully I'll remember this GIF and it'll come back when we actually look at the cardiovascular system, because it's a pretty good one. Okay, so the 3 types of muscle tissue: skeletal, smooth, and cardiac. So we're going to go through each one, look at the unique characteristics of each tissue, and so then hopefully at the end of this you'll be able to compare and contrast them. So look at the things they have in common and the things that they have that are unique, or the combinations of characteristics that are unique. So we'll start with smooth muscle fibers. Smooth muscle fibers have this grouping of characteristics. So the cells have a spindle shape, which is called fusiform. So what that means is that they are widest in the middle, and then they taper at each end. They typically have a single central nucleus. They are not striated, which means that they don't have stripes. And that's relevant because the other 2 types do. And they're involuntary. And when something is involuntary, that means it's controlled by part of the nervous system that we're not aware of. So we don't have any conscious control over our smooth muscle. Sorry, stuff in the way. Now, we find smooth muscle in a lot of places. It's a pretty widely distributed tissue type. And so the general location is in the walls of hollow organs. So that includes blood vessels. It includes the intestinal tract. It includes the reproductive system, quite a lot of it. Urinary system. Basically anything where we have hollow organs, we're going to have smooth muscle in the walls. That's not the only location, but that is the major location. Everything else kind of is an exception. Now, one of the exceptions we learned about already, so the erector pili muscle that is associated with each hair follicle and gives us goosebumps is a smooth muscle. And that's why we don't have direct control over it. We can't decide to have goosebumps. We just do or we don't, because they are involuntary. Okay, so that's smooth. Here's some pictures. So you can see this is a picture showing you that fusiform shape. And then all these little lines are the proteins that allow it to contract. So when these cells contract, they kind of like get very squished up and they almost like twist together. And so they can shorten quite substantially. And then this is just showing us how it's controlled, and we're not going to worry about it too much right now. We'll get to that more when we get to the nervous system. And this is a picture of it. So each of these little purple things is a nucleus. And we can't really see the outlines of the cells, but they're kind of like this.
Wow.
And then this is showing you how vascular it is, because these are actually blood cells right here in a little blood vessel. All right, whoever's got their mic on, if you're not asking a question, would you mind muting? Because we're getting a lot of noise. Excellent. All right, second muscle tissue type, cardiac muscle fibers. So I'll start with the easy part. This stuff only in the heart. Only place we have cardiac muscle tissue is inside our heart. So the characteristics, heart muscle cells are striated, and that means that they have stripes, which we'll look at in a second. They also have a single nucleus, which is similar to the smooth muscle. Instead of being fusiform, they have this weird branchy shape. And the branches are part of how they connect to other cardiac muscle cells. So they kind of do this. So they're really well connected to each other. And the connections between them are called intercalated discs. There's a couple of special things at the intercalated discs. So there are gap junctions, which remember are the little holes that electrically connect the cytoplasm of cells. And then there's desmosomes. And the desmosomes hold all the cells together so that they can't get ripped apart. Because heart muscle is always, just like that GIF I showed you, it's always contracting and relaxing and expanding. And so it's under a lot of pressure when it does that. So the tissue could be damaged if they weren't really well connected to each other. So we have these gap junctions and desmosomes in between each cell's connection to the next cell to keep everything really well connected. The gap junctions, by electrically connecting these cells, allow them to behave in unison. And we'll talk more about that when we get to the cardiovascular system. But there's a fancy name for it when a bunch of cells are able to work as one, and it's functional syncytium. So just to introduce some challenging vocabulary there. The other thing that cardiac muscle tissue has in common with smooth muscle is that it is also involuntary. So we have no conscious control over our heart muscle. We can't decide to make our heart beat or not. It just is doing it on its own outside of our control. So got some pictures. So here's some pictures. Diagram here. So you can see the intercalated disc is very, it's got a lot of little pegs, so they really interlock together quite strongly. And we can see them under a microscope as well, showing us those. And I think I've got a close-up, there we go, showing us the gap junctions in the intercalated discs, as well as the desmosomes. And you can see these are mitochondria in these cells as well. And then the other thing I want to show you is the striations. So as you're looking at this, this is a couple of cardiac cells, fairly close up. So these darker blobbies are the nuclei, one per cell. And then these dark lines are intercalated discs. But then hopefully you can see there are some thinner lines that run in a regular pattern that give these cells a striped appearance. And this striping is what we call striations. And the striations are a result of the contractile proteins in these myofibrils. And we're going to talk about that in just a little bit. Okay, so striped means that there's these myofibrils packed inside the cell. third type of tissue, skeletal muscle fibers. These guys are by far the weirdest, most different type of tissue. So first of all, the cells are really, really long. And we say they're cylindrical, but they're so long that they're basically like spaghetti or ropes or something like that. And they're multinucleated. So there's lots of nuclei inside of a single cell. Like cardiac muscle, they are also striated. So the whole cell is striped. I'll show you a picture. And unique among our muscle tissue types, this type of muscle is voluntary, which means we can have conscious control over it. So this is the only type of muscle that is voluntary. And just like the heart, we have specific locations for this. So the only place we have skeletal muscle fibers is in skeletal muscles. And most of them are, of course, attached to bones so that they can move our skeleton. So some pictures. We're going to look at this picture again later because this is our major diagram. But this is just a little section of a skeletal muscle cell. And it's packed full of myofibrils in here. This is showing you the striations and the multiple nuclei. And the reason I say it's just a section is because they are really, really, really long. And so it could be, you know, this long, and then they just cut a chunk out of it to show us. So we're going to look at that in a little bit more detail because pretty much the rest of the stuff we cover in muscular system is going to be referring back to skeletal muscle fibers. And that's because cardiac muscle, although it's a muscle tissue, is in the cardiovascular system, and smooth muscle really belongs to all of the systems in which we find smooth muscle. So this is the only system that we have skeletal muscle in, so that's what we concentrate on as far as looking at greater detail of anatomy and physiology. Should have, yep, there's a picture. Okay, so these are some fibers. So again, hopefully you can appreciate the striping pattern of striations that tells us the myofibrils are there. The big difference here when you're looking at this versus that cardiac muscle is that the nuclei are squished to the edges instead of being able to hang out in the middle. Does anybody have any questions before we move on from tissues? All right. Here's another picture, less magnified, but you can really see the striping in this one. I appreciate why we say that that's one of their characteristics. So this one is just giving you all of them, so it makes for a nice review. So make sure that you feel comfortable both comparing and contrasting our three muscle tissue types as far as features and locations. I thought I removed this slide, but apparently I did not. Okay, so the next thing we're going to start working through is understanding how muscle cells contract, so how these myofilaments actually result in forceful shortening of our muscle cells. And although we're going to focus on skeletal muscle, this is basically the same way it works in all three of our muscle tissue types. So in order to understand the contractile nature of muscle cells, we need to look at some of the specializations that we see in the cells. So we learned our basic cell anatomy before, and now what we're going to see is that when we have highly specialized cells, sometimes the anatomy doesn't stay the same, sometimes it becomes specialized too, and so we give it different names. So for example, the plasma membrane of a muscle cell is called the sarcolemma. Sarco is one of the prefixes we use when we're talking about muscle. So we're going to have a lot of sarco things today. The sarcolemma is specialized in that it has internal extensions. They're kind of like tunnels that start on the outside of the membrane and they go in. We'll see a diagram in a second. And so those extensions are called T-tubules. So these are the things that are special about the membrane. The endoplasmic reticulum has also been highly modified. Its primary job is actually to store calcium. So because it's been highly specialized as part of the muscle cell, we call it the sarcoplasmic reticulum instead of the endoplasmic reticulum. And it's much more extensive than it is in most other cells. And it has these enlargements at the end, like the edges, that hold like most of the calcium. And so we call those terminal cisternae. So a cistern is basically like a really big jug that can hold water or some other liquid. And when something's terminal, it just means it's at the end. So the terminal cisternae are large storage things at the edges of the sarcoplasmic reticulum. And again, I've got a picture, I'll show you in a little bit. And then of course, the major difference between muscle cells and other cells is that they have these myofibrils that I've already mentioned. And especially in skeletal muscle, that's the major thing in the sarcoplasm or the cytoplasm, because we of course give that another name too. So just packed full of these myofibrils, which are bundles of contractile proteins. So here's that same diagram. So sarcolemma, specialized membrane. This picture doesn't have the T-tubules on it, so I will show you that in a little bit. But the nuclei are shoved to the outside. So we say they're peripheral nuclei. They're basically just like shoved right underneath the sarcolemma. The mitochondria are arranged in rows in between these myofibrils. So when we look at it along the length, we see like a bunch of mitochondria in between the myofibrils. And then when we look at these T-tubules and terminal cisternae of the sarcoplasmic reticulum, we see that they all come together. And so the organization of both of those things is called triads, which I'll show you on the next slide. And that's going to be important when we look at the physiology of muscle contraction. So here's a close-up of the membrane. So these are the holes that are the openings of the T-tubules. And so then we see how the T-tubule comes down and they all kind of just wrap around these myofibrils. So that's a myofibril, that's a myofibril. And then all of this other spiderwebby looking stuff, this is the sarcoplasmic reticulum. And these are the terminal cisternae. So we have a T-tubule and a terminal cisternae on either side. And that combination is called the triad. I don't know why, but it is. And hopefully once we get through the physiology, you'll understand why I'm pointing that out. All right, so this one's just a fancier picture and I like it, so I show it. This one gives you a better view of how the mitochondria are kind of packed in rows in between the myofibrils. And then it's got a nice little peripheral nucleus here. It's got the T-tubule openings. And then there's a nice close-up where you can see the triad, T-tubules, terminal cisternae, and how web-like this sarcoplasmic reticulum is. So I really want you to appreciate the fact that the sarcoplasmic reticulum and the T-tubules completely surround each of these myofibrils. And the key here is that that's going to ultimately provide a source of calcium to the myofibrils. So it gets stored in here and released to the myofibrils, and that's going to have an important consequence when it comes to muscle contraction. All right, so let's look at the details of the myofibrils. So I guess I should have added this before. Myo is the other prefix we use when referring to muscle. So sarco and myo both just mean muscle. So a myofibril means like a muscle thread, and then a myofilament, which are the protein strands that make up the myofibrils, just means like super tiny muscle thread. So there's two types of these myofilaments. There's thick and there's thin. And they're arranged in parallel, which means that they're running longwise with each other. And when we pack a bunch of them together, we end up with those myofibrils that we've been looking at already. And of course, we pack a bunch of myofibrils together and we get the inside of a muscle cell or muscle fiber. So I've had it written up there, but I don't think I actually said it. So skeletal muscle cells are called muscle fibers because they're really long and skinny. So just like when we have, you know, some sort of material that we can make into a cloth, we call each individual strand a fiber or a thread. We call these cells fibers as well. And then because we call the cell a fiber, we have to use modifiers to mean smaller threads. So we got fibril, and then we got filament. So they just keep getting smaller, but they're all really, really long and thin at different levels. Okay, so thick filaments are up first. So thick filaments are made of a protein called myosin. So a myosin is this weird thing that looks like two golf clubs put together with the handles of the golf clubs twisted. When you take a bunch of these funny golf clubs and you stack them together, you get a thick filament. So there's a couple of characteristics of these myosin proteins. There's two heads on each one, and the heads are able to bind ATP, and they're able to bind another protein called actin. And then this is a hinge region. It's called a hinge because it's capable of movement. And then the rest of it is these two tails. And the two tails are basically just structural support. They maintain the myosin's position in the whole filament. So then we have thin filaments. Thin filaments are actually made of three different proteins. The most important one is actin. And actin is ultimately the protein that interacts with the myosin. So actin has a myosin binding site. And then these other two are called regulatory proteins because they control whether or not actin is able to bind to myosin. So troponin is the protein that is in control of the thin filament. It's capable of binding to calcium. This is where calcium comes in. And it also can bind to the two other proteins in the thin filament, tropomyosin and actin. Tropomyosin is the protein that can block the binding site on actin for myosin. So let's see what that looks like. So actins are these little round guys. I don't know why they draw them in green, but it makes them look like little peas. But they're basically set up like a two-stranded pearl necklace where they're running together. So each of these little guys is an actin, and it's got this little binding site for calcium. But the binding site is covered by these strands of tropomyosin. So tropo because it's being controlled by troponin, myosin because what it's blocking is actin's ability to bind to myosin. And then the troponins control the position of the tropomyosin. So when there's no calcium, tropomyosin blocks the binding sites. When troponin binds to calcium, it moves the tropomyosins, and now actin is able to connect to myosin. Okay, so thin filaments have two states. There's this one where everything is covered, and then there's this one where they're able to interact with the thick filaments. Now, when we put them together into the myofibril, we see that they're not just running together in a haphazard fashion. They're highly organized so that we have a bunch of thick filaments that are sitting together here, and then they're partially overlapped with thin filaments that are also running together. So this organizational pattern is called a sarcomere. The sarcomere is the functional unit of the myofibril. So remember from bone, where we learned that compact bone has a functional unit called an osteon, where that's as small as it gets and still able to be the thing, right? So if you break down bone tissue smaller than an osteon, it's no longer recognizable as compact bone. If you break down these filaments smaller than a sarcomere, it's no longer able to perform the function of contracting. So let's look at this sarcomere anatomy, because this lets us understand how contraction of muscle fibers occurs. All right, so this is just a different diagram, and it's showing you the thick filaments lined up in the middle of the sarcomere, thin filaments overlapping on either end, and they're connected right here. And this overlap is important because the overlap is the place where the myosins are able to bind to the actins when that binding site is exposed. And what we see is that the myofibril itself is just a length of repeating sarcomeres. So that striped appearance we get is from these repeating patterns of thick and thin filaments along the length of the myofibril. Now, the reason why it's a light and dark striping is because the thick filaments create these dark bands, which they named A bands because of the way they discovered them. And then when we don't have any thick filaments, where it's just thin filament, it looks lighter, and those are called I bands. And so that repetition of thick and thin filaments down the entire length of the myofibril gives us this striation. So A band is defined as the length of the thick filament in the sarcomere. It's also the middle of the sarcomere. And then the I band is the length of the thin filament in the sarcomere where there's no overlap with thick. And the reason why we define it as only the portion of the thin filament where there's no overlap is because once we get overlapped with the thick filament, it's going to look dark, and so it's going to be the A band. Now, the interesting thing about these I bands is that really when you look at the sarcomere, the A band's in the middle, and then part of an I band is on either side. But when you look at a myofibril, we see that it just looks striped. And that's because these sarcomeres are end to end, right? So the next sarcomere is over here. And so we typically have half an I band for one sarcomere, half an I band is the other sarcomere, and that's how we get this alternating stripe pattern, even though we have I band on either side. Now these anchors on the ends of the sarcomere are called Z discs. You'll also see them called Z lines. And so these are additional proteins that the thin filaments are bound to and they mark the edges of the sarcomere. They don't show up in the striping, but we know that that's where the edge is. So let's look at this guy again. So again, we've got this A band is the whole length of the thick filaments. Here are our Z discs, and I do believe that they do have this patterning of zigzagging, which is where they got their name from. And then however much thin filament is not overlapping with the thick is the I band. Now there are other things that we label in here, but you don't need to worry about them. Now this one is the same thing I just showed you, but three-dimensional. And the reason why I put a three-dimensional one in there is because this is how it looks in reality. So it's, even though we represent a sarcomere as a very flat thing, as we're learning it, we know that these myofibrils are round, and that means that the arrangement of the thick and thin filaments is also going to basically be in a cylindrical pattern. So when we look at this, this is also a sarcomere, and these are the Z discs on the end. This is the length of the thick filaments, which makes this the A band. And then we've got I band on either side, where the thin filament is not overlapping with the thick. Oops, sorry. Okay, so can everybody see that that's the anatomy of a sarcomere? You guys can do a little check, your participation, or, thumbs up. Yes, typed in is also acceptable. Okay, so if we can understand that anatomy, then we can apply it to the physiology of contraction. So the way that the whole muscle cell shortens is because the thick and thin filaments actually slide past each other. So we call it the sliding filament model. And literally what happens is the thick and thin filaments increase the amount of overlap they have so that the entire muscle shortens. Because if every single sarcomere is doing that across the entire length of the myofibril, then it shortens the entire muscle cell. So contraction then isn't about shortening of a protein, it's about sliding proteins over each other. And the way that we do that is by binding actins and myosins together. Binding actins and myosins together is called cross-bridge formation, which we'll talk about in a second. And so when we look at contraction defined at the level of the sarcomere, we see that contraction is actually just activating that binding process. And if that binding process goes on for long enough, then we'll actually get to shortening. So shortening is when we actually achieve what we call muscle contraction. And it does that by increasing the overlap between the thick and thin filaments, as I already said. Oh, I forgot to take that out. Don't worry about that. Now the A band never changes because it's defined as the length of the thick filaments. But we will actually see a decrease in the I band because remember the I band is just the thin filaments where there's no overlap. As we increase the overlap by bringing the sarcomere together, we lose some of our I band. So this is showing you another diagram of a sarcomere. Now this is actually a picture of a sarcomere under a very high magnification. And so this is the A band. So these are the thick filaments. These are the Z discs. And these are the I bands. So it really does look like that when you look at it under a microscope. And there we go. So we can look at the proportion of A band to I band in a relaxed sarcomere, so one that's nice and lengthwise. And then when we have contraction and shortening, we can see that, so here's the Z discs, we can see that we have decreased the length of the sarcomere, but we haven't changed the length of the A band because we don't change the length of the thick filaments. Instead, we just lose a lot of I band because we've increased the overlap between the thick filaments and the thin filaments. So this is, once they were able to get to this level, we were able to confirm that the sliding filament model is actually what happens. And so we know that when muscle contraction and muscle shortening occur, it's because we are overlapping our thick and thin filaments. Does anybody have any questions? I'll give you a second to think about it. Okay, so if you're very visual, there are videos that show this happening and also show the physiology stuff that we're going to talk about next. So these are the different physiological events that occur in muscle contraction, and this is sliding filament. Now there might be better videos. These are the ones that I found a while ago. If you find a really good one that you love, please share it on a discussion in class, or on our class Canvas site, and I can update this later. I do find it useful because this is literally stuff that's moving, and so it's easiest to see when it's moving. Okay, so before we get into the physiology, I want to address the gross anatomy. So this is the anatomy of actual muscle organs, as in what happens when we put a bunch of muscle cells together, plus all the connective tissues and blood vessels and nerves and stuff that go along with it. So just like bone organs are bone tissue plus a bunch of extra stuff, muscle organs are the same. So we have a bunch of skeletal muscle tissue. Just a reminder that this is always skeletal. We're going to have a bunch of connective tissues holding everything together, and we'll talk about them in a second. There's always going to be a nerve associated with the muscle. That's how there's control over muscle contraction. And then remember, these are always vascular, so there's always a fairly large artery and 2 to 3 veins for each muscle. So when we put all of that together, we get a fairly organized organ with 3 distinct layers of connective tissue. And this MYS is basically the same as the myo, so mesium because it's around the myo. Okay, so the epimesium is the outermost connective tissue layer. It surrounds the entire organ, and it's going to be a fairly dense connective tissue holding everything together. And then it's got these inward projections, so you can kind of think about it like an orange peel. And then you know how the orange is divided into these sections? And the white pulpy stuff comes in from the outside. So it has these inward projections that divide the muscle into groups or compartments. Those are called fascicles. And the connective tissue surrounding each fascicle is called the perimesium. So peri for perimeter. And then each fascicle has a bundle of muscle fibers, the individual cells. And each individual cell has just a little bit of connective tissue around it. So we call that layer the endomysium, endo for internal. And this is just a little bit of fine areolar tissue. It just helps all the teeny tiny blood vessels and teeny tiny nerve endings have a pathway to get to each individual cell. Any additional connective tissues around an individual muscle are going to be called fascia. So fascia is connective tissue separating organs or layers of tissue from each other. And so, for example, between skin and muscle in our limbs, we'd have some thin layers of connective tissue fascia separating the hypodermis, that adipose layer, from the epimysium of each muscle. So let's see what that looks like on our diagram. So this is a diagram of an entire muscle organ, okay? So the epimysium is this bit that they've sort of flapped back surrounding it. And muscles tend to end in tendons, which is what they're showing down here. So the epimysium is going to be continuous with the tendon, and there's probably a bone here that it's connected to. And then you can see the connective tissue coming in. That's the perimysium. And each of these bundles is a fascicle. Ooh, that was not well written at all, but anyway, it's right there, muscle fascicle. And then each fascicle has a bunch of individual cells. And surrounding each cell is going to be that endomysium. So it's this guy here, they're showing you close up. It should not be boxed like this. It should be boxed like this. And then this guy is this guy that's sticking out. So this is a fascicle surrounded by perimysium. And then we've got epimysium around each of these individual cells. And then this cell is showing us that we've got a cell packed full of myofibrils, peripheral nucleus, right there. And then mitochondria and some nuclei. And at each level, one of the reasons why we have these connective tissues is because we have blood vessels and nerves running through. So there's going to be blood vessels and nerves entering through the epimysium, running in the perimysium between the fascicles, and then entering each fascicle to get to each individual cell. Okay, so one of the functions of these connective tissue layers is to organize the blood vessels and nerves for each muscle organ. All right, any questions about the anatomy? So that's microscopic anatomy and then gross anatomy. Okay, so we are going to at least get started on our muscle physiology. We've got about half an hour left, so we'll see how we go. So this is the complicated part, which is how this actually works. And I tried to cut it back because I usually, like I said, I usually teach a 2-semester course, and obviously we can't cover the same level of detail in a 1-semester course. But you know, I had to have enough detail in there for it to make sense. Before we do that, give me 1 second. I do not know why the dog will not stop barking. I'm sure you can hear that. So let me just pause the recording and I'll go see what's going on. Okay, so now we're going to jump into the physiology of how we actually get that sliding filament model to work. Okay, so we're going to go through a couple of different physiological steps, and hopefully I didn't put too much detail in it. The first thing we want to look at is how the nervous system is able to control the muscles. And so we need to understand a little bit of the things we've talked about before, which are ion channels for the movement of electrolytes into and out of the cell. We're mostly going to be concerned with sodium and potassium. We're going to touch back on that idea of arresting membrane potential and that electrical charge across the membrane. And then that primary active transport example that I told you guys about before, the sodium potassium pump that uses ATP to move sodium and potassium against their concentration gradients, that's going to play a really important role here. So we're going to touch on those things that we've talked about before, and then we're going to apply them to this physiological process. So there's actually a couple of different types of ion channels that are relevant for this process. There are leak channels, those are always open, and there are potassium leak channels that are always open, allowing a little bit of potassium to leave the cell. This leakage allows the inside of the cell to normally be negative, which I'll talk about more in a second. And then there's two types of channels that allow the electrical activity of both the nerve and the muscle. So ligand-gated channels, ligand just means that it's able to bind something, and then binding of something opens or closes the channel. In this case, it's going to open it and allows something to move through, which will be ions. And then voltage-gated channels open and close in response to electrical changes. And that's going to be a major factor here. So remember that the sodium and potassium pump is kind of our classic active transport example, using ATP to move sodium out of the cell against its concentration gradient and potassium into the cell against its concentration gradient. So this allows the cell to maintain a high level of potassium and a low level of sodium. And in combination with the leak channels for potassium, oh boy, sorry, the handwriting's getting bad, is what maintains arresting membrane potential. So arresting charge across the membrane. So here's a diagram of that. This is the pump. This pump is using ATP to send sodium out and potassium in. And then as we leak a little bit of potassium back out, we end up with this negative charge just inside the membrane and a positive charge just outside of the membrane. Remembering that the entire cell is neutral and the extracellular fluid is neutral, this charge is just on either side of the plasma membrane, and it works very much like a battery. So the resting membrane potential then is established by those things. And these are just the words showing you what I already said. Okay, so the electrochemical gradient is what we just saw for the pump. So it's the charge difference and the sodium and potassium concentration differences. So here's another one. It's showing you basically the same thing, but now it's sideways. And so again, we were a little bit negative on the inside, a little bit positive on the outside. I was debating whether to leave this in or not, and I was like, oh, sure. And now I feel like it was redundant. Okay, so you don't need to know the number, but just because it's going to be on our graphs. Most of the time, our resting potential is negative 70 millivolts. Okay, so it's a little bit negative because we're pumping more sodium out than potassium in, and the potassium is leaking out, so we have that negative charge. And when we have a charge, we say that the cell is polarized. So polarized means that we have a resting membrane potential. And I point this out because we're going to use that word as we talk about changing the charge of the membrane. All right, so when we change the charge of the membrane, we want it to be really, really fast. And that means we need to open enough ion channels that a bunch of ions can passively move down their concentration gradients in order to change the charge in the membrane. So if we let a bunch of positive stuff into the cell, we're going to very quickly change the charge so that it becomes positive. So because we've changed the charge, we call this depolarization because we're removing that polarized state. And the way that this happens in muscle cells is because there are voltage-gated sodium channels. So sodium channels that open once there's already a change in charge. So let's look at that in a diagram. So this is resting on the left. And then when a stimulus, and we'll talk about what that stimulus is in a little bit, when a stimulus opens gated sodium channels, it lets a whole bunch of sodium rush in. Because sodium is positively charged, this changes the charge inside the cell to positive. And because a bunch of positive stuff left outside the cell, now that's negative. That part doesn't really matter. What matters is that the inside of the cell is positive, and we call it depolarization. So that gets us a change in electrical activity. And we'll talk about the consequences of that in a little bit. So that's kind of the thing that makes stuff happen. But then we also need to be able to reset, because we need these excitable cells to do this over and over and over again. So in order to get back to resting, which is resetting the cell, we have to then reverse this depolarization event. And the way we do that is by opening more ion channels, and usually they are also voltage gated, and allowing more ions to go down their concentration gradients to rapidly reverse the charge again. So what usually happens is that now we let a bunch of potassium out. And as these positively charged potassiums leave the cell, we end up with a negative charge inside the cell again. So because we're returning the cell to a polarized state, we call this process repolarization. So let's go back to that diagram. So now we are letting potassium move out in a large volume, and that positive leaving the cell makes the cell negative again. Now, in order to accomplish that, the other thing that has to happen is that those sodium channels have to close so that that's not happening at the same time, okay? So we started at negative, we depolarized to positive, we repolarize back to negative, and now we have to reset everything, because now we have a bunch of sodiums inside the cell and a bunch of potassiums outside of the cell, and that's not where we want them to be, okay? So this is what it looks like when we graph it. So this is resting down here. This is when nothing's happening. A stimulus is going to change the charge. And we'll talk about what that is in a little bit. So this is the point at which we're opening voltage-gated channels, and we're going to get a bunch of sodium into the cell, and that's going to get us depolarized. And then those close, and potassium channels open, and potassium leaves the cell, which takes us back to negative. And then those sodium-potassium pumps have to reset everything and get us back to resting. So they pump all the sodium back out and all the potassium back in and get us back to where we were before. And once that's done, then it can go back and do this all over again. This is a purely rechargeable battery. Does anybody have any questions about that? So now we're going to look at how that electrical activity occurs and how it results in actual sliding filament overlappy stuff. Okay? Until we run out of time. So the way that we control this is with a neuron, right? So the nervous system controls the muscular system, and nerves are just a whole bunch of neurons. So the neuron is the nervous system cell that controls other things. And we'll talk more about that when we get to the nervous system. The type of neuron that controls skeletal muscle is called a somatic motor neuron. So somatic means body, and generally when we're talking about muscles and bones and stuff, we use somatic. And that's as opposed to when we talk about internal organs, like, you know, the heart, lungs, digestive system, and then we use visceral. So somatic and visceral are opposites. So somatic tells us that it's controlling like the musculoskeletal part of the body. Motor tells us that, you know, it's doing motor stuff, and neuron just tells us it's a neuron. So one motor neuron is going to control each muscle fiber. Sorry, that should say neuron, not nerve. And then that neuron actually controls more than one muscle fiber. And we'll talk more about that on Wednesday. So motor neurons have multiple muscle fibers to control, but each muscle fiber only ever gets its stimulation from a single neuron. So there's never multiple things telling it what to do. There's always only ever one thing telling it what to do. Now, the point of control, the place where the neuron comes in contact with the muscle fiber, is called the neuromuscular junction. So a junction is where more than one thing comes together, and neuro for the neuron, muscular for the skeletal muscle. Any place where a neuron is controlling something, even if it's another neuron, is called a synapse. So this is also a synapse, but the particular one that it is neuromuscular junction. So neuromuscular junction then specifically refers to a synapse between a somatic motor neuron and a skeletal muscle fiber. Now, because neuromuscular junction is a mouthful, we like to abbreviate it to NMJ, especially when we're writing it down. So we don't have to write out neuromuscular junction every time. So you'll probably see me use that abbreviation. And it also has another name. I don't expect you to know it, but if you do more of this stuff later, this might show up because this is a synonym. It's also called the motor end plate for whatever reason. So this is showing us a neuron coming down to synapse with this muscle cell, and we're going to talk about what happens there, how electrical activity in the neuron results in electrical activity in the muscle. So first, a little bit more anatomy of this synapse. So remember, the synapse is also the neuromuscular junction. So even though the neuron has electrical activity and the muscle cell is going to have electrical activity, we can't have the electrical activity go straight from one to the other. They're not electrically connected. There's actually a little bit of a space. And so instead, there's a chemical connection. And that chemical connection is bridged by a neurotransmitter. That's how neurons control almost everything, is that they use chemicals called neurotransmitters. And in this case, that neurotransmitter is a chemical called acetylcholine. So the muscle fiber has receptors for the neurotransmitter, so acetylcholine receptors. And surrounding the synapse is an enzyme that can break down that neurotransmitter, acetylcholinesterase. Now, because again, this is a fairly long word, we abbreviate that ACH, and then this enzyme can be abbreviated ACHE. The reason we want to be able to break it down is because we don't want it to last forever every single time a neuron releases some acetylcholine to a muscle, right? We need this to be able to be turned on and then turned off again. All right, so that's acetylcholine. So we can look at this anatomy like this. So here's the end of the neuron. It contains a whole bunch of acetylcholine. And when electrical activity travels down the neuron, it causes the release of the acetylcholine. So this nerve impulse, which is also called an action potential, is the electrical activity of the neuron. And then acetylcholine gets released from the neuron and it travels over to the membrane of the muscle cell, where it binds to these receptors for acetylcholine. When it binds to the, when it binds to these receptors have a change in them. So these receptors are also ligand-gated channels, which means that they can allow ions through when a chemical binds to them. And in this case, the chemical is the neurotransmitter acetylcholine. So acetylcholine binds to these receptors. These receptors then are able to allow sodium to move into the cell. Did I write that down? Yes. As well as potassium. So at this neuromuscular junction then, we have a depolarization event. And that depolarization event is enough to open additional channels, voltage-gated sodium channels, in the rest of the muscle cell membrane. So now this muscle cell is opening voltage-gated sodium channels, just like I showed you before, and that's depolarizing the cell. So we're getting that depolarization. And then it'll also open potassium channels after that happens. So then we'll get a repolarization. So now what we get is electrical activity of, let me go back before I get to that. Electrical activity of the neuron has now triggered electrical activity of the muscle fiber, even though there was no direct electrical connection. Okay, so acetylcholine bridges the gap between the two cells and causes the electrical activity in the muscle fiber. And it moves through just like we talked about. So then we have that repolarization, voltage-gated potassium channels open. The sodium channels do close at the same time, so that restores that negative charge and repolarizes. And then we have to reset the cell, right? So remember, then we have to get those sodium-potassium pumps to restore normal conditions. And during that period of time, the cell cannot be depolarized again. So we can't get another electrical event during the period of time where it's resetting. And so we call that period of time the refractory period, where the membrane is resetting itself to normal resting conditions. And then once it's reset, it can be stimulated to depolarize again. So this is what it looks like. Another graph with a little bit more detail. So now we know that the stimulus is the events at the neuromuscular junction causing acetylcholine to open these ligand-gated sodium-potassium channels, and that's going to trigger the opening of these voltage-gated sodium channels. We get the depolarization. The sodium channels close. the potassium channels open. That gets us repolarization. And then this part is the refractory period. So this is where we reset and the cell isn't responsive. And then when we get back to resting state, then we can go back and do it all over again. So again, this is also called an action potential. And when it's occurring to the muscle cell, we also call it a muscle impulse. So this will occur as long as the neuron ending is releasing acetylcholine, and it'll keep releasing acetylcholine as long as it's getting an electrical signal. And that's function of the nervous system, which we'll cover later. But once fresh acetylcholine isn't being released anymore, we can stop the signal because the enzyme, the acetylcholinesterase, is going to break down the neurotransmitter acetylcholine so that it's no longer binding to those receptors. And this is a far more detailed slide than you need, but I just wanted a picture that had acetylcholinesterase in it, breaking down the acetylcholine. Okay, so that stops the nerve impulse, and then that stops the electrical activity in the muscle cell as well. Okay, does anybody have any questions about what happens at the neuromuscular junction? So how the neuron excites the muscle cell. Going to assume that means we have no questions. Okay, so one more thing before we run out of time. So you know that the sliding filament model causes contraction, right? We're overlapping. And you know that calcium has to be involved. And so the way that the electrical changes in the muscle cell membrane results in the ability of the sarcomeres to overlap is called excitation contraction coupling. So we gave it this really fancy name, and we abbreviated that to ECC. But basically, what happens is the electrical activity, the depolarization of the membrane, depolarizes the entire membrane, including the T-tubules, which remember go deep into the cell and wrap themselves all around those myofibrils. And they're hanging out right next to the sarcoplasmic reticulum terminal cisternae, which are storing a bunch of calcium. So when T-tubules depolarize, it opens channels that release the calcium from the sarcoplasmic reticulum into the cytoplasm of the cell, and that allows a bunch of calcium to flood into the myofibrils. So the calcium surrounds these myofibrils and binds to, sorry, this should say troponin, binds, hate when that happens. I knew there was a typo in there and I couldn't see it before. So it binds to the troponin. The troponin allows the tropomyosin to move off of the actin, and now the actin is able to bind to the myosin, and that allows the sliding filament model to occur. So here's a better detail about the T-tubules. So when we have depolarization happening here, it also happens all the way down here. And as this depolarizes, this releases calcium. So once we release calcium, we can have that sliding filament model take place. and we call that the contraction cycle. So we'll start up on Wednesday with the contraction cycle so that you have time to process this. And that way, if you have any questions about any of the physiology, we can hit it at the beginning of lecture on Wednesday. Anybody have questions right now? Nobody? Okay, so I am going to try to get you a little practice test up so that you can try out the formatting of the test through Canvas. And I'll try to make you another worksheet for joints and make you one for muscles as well. They're taking longer than I thought they would. And so that's why you've got what you have. If you've seen it already, there is one worksheet. It looks like a quiz, but it's not a quiz. If you haven't seen that yet, just let me know. I can show it to you if you didn't understand the announcement I sent. So no questions? All right, let me stop.